So I start with the DNA fingerprinting. Yesterday, we started this, but I'm going to repeat it for you. So the first step in DNA fingerprinting is source of DNA. So first is source of DNA, which we get from the crime scene, or which is the sample which we collect uh, for the process of DNA fingerprinting. So the first step is source of DNA. And what is the source of DNA? How much sample is collected? So of tissue, or 1 lakh of skin, and these can be of hair. So many cells are collected, 1 microgram of tissue, or 1 lakh cells are collected. These are taken as samples. So these are taken as samples, collected as samples. Now, mainly what happens, the samples which are collected from the crime scenes are very less in amount. So what needs to be done to amplify? That means we need to increase this in amount, and this is done by one of the technique. So this will come under the topic DNA amplification if the sample is less in amount. So it is amplified. DNA amplification. This is done by a technique which is known as PCR, or polymerase chain reaction. And what is done in this, the small amount of DNA is amplified. That means many copies are made if it is less in amount. Many copies of the sample collected are made if it is less in amount. So if it is less in amount, then what is done, that it is amplified by using the PCR technique, and we get sufficient amount of the sample, which can be processed for the process of DNA fingerprinting. Then the next is, what we will do. The next is extraction of DNA. We have collected the sample. Now it is with the cell. That means it is, the DNA is not been, has not been taken out. Still the DNA is in the cells, is in their cells. That means the DNA needs to be taken out. Right now, only the samples are collected, and still the DNA has not been extracted. We have collected the sample because the sample was very little. We have amplified the sample, but still the DNA, the thing which we want for the process of fingerprinting, is not there. That means we do not have DNA right now. We have the complete cell. What do we want? We want DNA. So that DNA from the cells needs to be extracted. So the third process is extraction of DNA. Third step is extraction of DNA. How this is done? This is done by using various, enzymes. Like this was what I was telling you yesterday when I finished with this topic. I was telling you that in class 12, this is an experiment to extract DNA, and I showed you the DNA, extracted DNA in a test tube, and that was the DNA extracted from onion. That was the DNA which was extracted from onion. So what I used the detergent or the shampoo. uh, Last year when we did this practical in class 12, so I used easy, and that was donated by Chitra. So okay, Chitra gave that. She brought it from her warden or somebody, that uh, easy which we used. And that easy, what it does, it breaks the cell wall, it breaks the membrane, and what we get ultimately is that DNA which comes out of it. So DNA is extracted by using these enzymes. So enzymes are used first to break the wall and the membrane, and later on what happens that the chilled ethanol means this ethanol is kept in the, this ethanol is used which is kept in the refrigerator for a long time, so it is chilled. So this chilled ethanol is put in the, this is this is this is what we poured in the test tube, poured in the test tube with the sample. This test tube has the sample. This test tube has a sample, whether it is blood sample or it is hair or it is skin or semen, whatever that sample. So when chilled ethanol is poured, DNA separates. DNA separates. It separates in the form of, excuse me. It separates in the form of threads. So it is taken out. It is pooled. Okay, it is taken out with a glass rod. Excuse me. So here DNA is separated. See, DNA gets separated from the complete content when chilled ethanol is poured into it. So finally we get DNA, and this DNA is further used for the process of fingerprinting. Now what is done? We have got a complete strand of DNA, and that long strand we do not want. We need to break it into small fragments. So next step is fragmentation. So what comes in the DNA fingerprinting? Next step is fragmentation. So fragments are made. It is broken down. DNA is broken down. The long DNA is broken down into small fragments, and the enzyme which is used to break this into fragments is endonuclease. So endonuclease. This is the enzyme. This is used to break the DNA into small fragments. That means what do we have now? We have different sizes of fragments of DNA which will be used further. So this is the fourth step in which fragmentation is done, in which DNA is broken down into small fragments by the help of restriction endonucleases. You can write down endonucleases, or you can also write down restriction endonucleases. Anything you can write down by the use of restriction endonucleases. Now what is done? After this, a technique is used. Right now you have the fragments, the pieces of DNA of different lengths. Now what do you want? You want to arrange them accordingly, and for arranging them according to their size, which technique is required? If anybody remembers, what is that technique which is required to arrange the various fragments of DNA according to their size? What is that technique? Yes. Yes, beta, what is that process? Yes, Paras, it's correct. It's electrophoresis. So next what is done is electrophoresis. So the next process is electrophoresis. And by the help of this technique, what is done? That the fragments are separated by this method and they are arranged according to their size. Fragments are separated and arranged on the agarose gel. Now you know what is agarose gel. Agarose gel is the plate on, this is the plate electrophoresis. On this, the gel which is spread is agarose gel. So that and fragments are separated and arranged on the agarose gel according to their size. According to their size. So this is done by electrophoresis. Now what happens? What is the charge of DNA? What is the charge present on DNA? Yes, what is the charge present on DNA? Yes, DNA is positively charged or negatively charged? Negatively charged. Yes, Amit Patel. It is negatively. Yes, Sukhsahaj, it is negatively charged. Now what happens? When electricity is passed through, the DNA fragments will start moving towards the positive charge. And when they start moving towards the positive charge, what happens that the bigger segments will stop first and later on the smaller segments. So we will get them arranged according to their size towards the extreme, towards the positive side. That means towards the extreme positive, we will have the smallest one. And towards this side, the other side, we will have the larger one. So they get arranged according to their size. That means when they move towards the positive side, they will get arranged like this. That means according to their size. So like this. So according to their size, they will get arranged. Now, this is how because of the method of electrophoresis, they will get arranged. So what is electrophoresis basically? That means by this method, the DNA fragments of different lengths can be distinguished. So what can be distinguished? DNA fragments of different lengths can be distinguished. DNA fragments of different lengths can be distinguished. Now what happens? The DNA is negatively charged. I'll write it for you. DNA is negatively charged. Therefore, when electric current is applied to the gel, which gel they are talking about here? They are talking about agarose gel. We can write to the agarose gel. DNA fragments migrates or start moving, anything you can write, migrates to the positively charged electrode. They start migrating towards the positively charged electrode. Then what happens? The shorter strands of DNA, they are more quicker, they are quicker, and they move quickly through the gel than the longer strands. So you can write down the shorter strands of DNA move quickly to the gel than the longer strands. Resulting in the fragments being arranged in order of size. So accordingly, in order of size, they get arranged. And then on this, when they are arranged on the agarose gel like this, it is very difficult to see them. because you cannot distinguish the various sizes of DNA on agarose gel. It is very difficult to distinguish. So what is done? There is a chemical name is ethidium bromide, which is a carcinogen. Ethidium bromide, mainly this is used, which is a carcinogen. It has to be used very carefully. Carcinogen. This is used to stain. This is used to stain DNA fragments to make them visible. So what happened? They are treated with ethidium bromide, which is a carcinogen, so that they are visible on the agarose gel. Otherwise, they are not visible. Then after this, what is done? See, now we have got the DNA fragments of different size on the agarose gel. Now this we have to keep in mind that agarose gel is not a stable substance. This is not a stable material. Very important. Agarose gel, which is present, which is on the electrophoresis. After electrophoresis, we get agarose gel. The DNA gets arranged on the agarose gel. So this agarose gel is not a stable material. That means if it is not used quickly, what will happen? All the DNA fragments, they will get destroyed. All the DNA fragments on agarose gel will get destroyed because agarose gel is not a stable material. Now, what do we want? Because these fragments of DNA can be stored for years. See, for years they should be stored, because say the DNA fragment of any extinct plant, which is not extinct yet, but in the coming time, if it is going to extinct, what do? We take out its DNA and we keep it. Now, if those DNA fragments of that plant which is going to get extinct, if those are taken in agarose gel, what will happen? In a short period of time, all the DNA will get destroyed. So we want such a material which is stable, so that the DNA fragments on that stable substance can uh, remain in a good condition for a long duration, for a long period of time. For that, another material is used, and that is a nylon membrane. That is used, which is a nylon membrane. So what is done? A more stable material which is used now is a nylon membrane. A more stable material is a nylon membrane. So that the DNA fragments can be stored for a long duration. Now, what is done? That the DNA which are right now present on the agarose gel, they are transferred to the nylon membrane, so that we can store that DNA for a long duration. Now, that process in which the DNA from the agarose gel is transferred into the nylon membrane is known as southern blotting. So the sixth step is southern blotting. This is the next step. And what is done in this step is that these VNTRs, we have taken only the VNTRs. Okay, variable number of tandem repeats. Those base sequences which are repeated a number of times, we have taken the VNTRs. Those VNTRs from agarose gel, these are present on agarose gel. But this is not stable. So these are transferred to nylon membrane. Okay, so these are transferred to nylon membrane, and this is advantageous to transfer it to nylon membrane because they can be stored for a long duration of time, and they will not get destroyed if they are kept in, if they are uh, stored, if they are transferred to the nylon membrane, because nylon membrane is a stable material and it keeps them in proper state, in right condition for a long duration of time. Now, after this, what is happening? Then the next step is synthesis of DNA probes. Synthesis of DNA probes. That means the probes are synthesized, the base sequence of which are known to us. We know that this particular probe, which has a DNA probe, has this base sequence. So what is done in this case is the radioactive segments of known sequence are synthesized. Radioactive sequence of radioactive sequence uh, segments of known sequence are synthesized. That means we take the segments and we know that what is the sequence of this. Suppose we have taken a segment and we know that in this segment we have this A, C, G, T, A, C, G, T. We know that this particular segment has this sequence, or it cannot. We take small of this. Suppose this small segments are taken. Suppose we have taken this, and we know that this radioactive segment, this has this sequence. So when we will put this segment into the bath in which DNA fragments are there, this will get attached to whichever fragment. We will come to know that this particular fragment has this base sequence. Okay, so the DNA radioactive segments of known base sequence, these are synthesized. After that, what is done is the next step after this is hybridization. So the eighth step after this is hybridization. What is done in hybridization is that the nylon membrane, now all the DNA fragments are present in the nylon membrane. This nylon membrane is immersed in a bath with DNA probes. So what has happened? Suppose this is the bath, and in this bath, all the DNA probes are there. Probes which we have made, the segments of known sequence. These in this bath, the DNA probes are there. Now what happens when we immerse this nylon membrane which has the DNA fragments? When we put this sheet, the nylon membrane, this, so whichever segment, whichever probe, where it attaches, we will come to know the base sequence of that, because this is of known sequence. Suppose it attaches here, this one attaches here. So what we will come to know that this particular segment has T C T G C A. We will come to know what type of bases are there in that particular segment of DNA, in that particular fragment of DNA which is there on the nylon membrane. So we can write down that the nylon membrane is immersed in bath containing DNA. So nylon membrane here, the next step, nylon membrane is immersed in a bath containing DNA probes. So DNA probes are there, they are immersed, so they will attach to their complementary fragments. When DNA probes attach to their complementary fragments. So they attach to their complementary fragments. So hybridization is done. Now we have come to know that the sequence of all of them. Now what is done? Finally, we have to see that where are those DNAs? Okay, so this sheet which has now probes attached to it, that is exposed to X-ray film. Last step, the sheet is exposed to X-ray film. Now, as already we have treated it with ethidium bromide, so wherever VNTRs are there, those will be visible in the form of bands. Those will be visible in the form of bands because of the ethidium bromide, what happens? They become visible. So when the nylon membrane containing VNTRs and probes are exposed to X-ray film, what will happen? That they will visible as dark bands. So when they are exposed to X-ray films, they will be visible as dark bands. Visible as dark bands. So this is going to happen during the complete process of DNA fingerprinting. That is, first of all, what we do that we collect the samples. After collecting the samples, what we do is we amplify the sample. That means if the sample is in less amount, it is made in sufficient amount by amplification by the process of PCR. And then after this, we extract the DNA. The DNA is extracted because still the DNA is present inside the cell. It has to be extracted. It has to be brought out of